proteins, but polyacrylamide gel electrophoresis can also be used to separate nucleic acids by size, whether to purify or analyze. So we can use polyacrylamide gel electrophoresis either in denaturing conditions such as with urea-PAGE or non-denaturing conditions to separate small pieces of DNA and RNA. And so today, let's talk about how it's done and how it works.
So one of the most fundamental techniques that we have in biochemistry is the ability to separate molecules by size using some sort of gel electrophoresis. And I'll get more into what the size refers to later. But with gel electrophoresis, basically this gel, you have this mesh, and you're going to send molecules traveling through this mesh. And the longer the molecules are, the bigger they are, the more they're going to get tangled up in that mesh. It's kind of like if you were to drop some jump ropes through a sea of basketball hoop rims. And so what's going to happen is they're going to get tangled up more. They're going to travel slower through the gel, and you get them to travel through the gel in the first place using electricity. So this is where the electrophoresis part comes in. You have a gel running box is going to set up this electric gradient where you have a positive charge at the bottom of the gel or at the end of the gel. So these gels can be run in like a slab format or a horizontal format. And the electricity, the charge gradient, is going to get the negatively charged molecules to travel through the gel towards the positively charged ends. So they're going to be traveling through, but the longer ones are going to get tangled up more. Because they're getting tangled up more, they're going to travel more slowly. So that when you turn off the electricity, you stop that gradient, you stop giving them the drive to move through. Now they're going to be stuck where they are. And when you go and you visualize where they are using some sort of stain or UV, you can then see the size of the bigger pieces are going to be higher up and the smaller pieces are going to be lower down. And then you can, maybe that's just all you wanted to do was just take a look, or maybe you wanted to actually extract the molecules out of the gel. And so you can extract them and then you've purified them from the other ones. But this isn't like, if we're doing this for protein, this isn't like protein chromatography. Like we're not going to be using those protein for things, but except for like spending them for mass spec or something. But when we're doing this for nucleic acids, we can isolate the RNA or the DNA away from the other things. And so you might have heard of PAGE purification, like maybe you're going to order some sort of a DNA or an RNA piece, and you have the option of PAGE purification. Actually, it's like PAGE purification, and you can do it yourself in the lab. You run the PAGE gel, and then you extract the piece out of it, and it's like concentrated and resuspended in a buffer and stuff. So I mentioned PAGE. So PAGE stands for polyacrylamide gel electrophoresis. So PAGE is one of the there's two kind of key gel types that we use in the lab. One is Agarose gel, which we're typically using for like big DNA pieces and also big RNA. It has a looser mesh. It's made up of agarose sugar, and it's good for separating really big things. And we run them in these vertical slab, these horizontal slab formats typically. There's also polyacrylamide gels. And so these are going to be made up of this polyacrylamide, and this is going to be a tighter mesh, a more consistent mesh size as well. You can also customize the amount of the meshiness, so you can make it add more of like cross linkers and more of the things like that to make it a tighter mesh, which is good if you're trying to separate smaller things, or you can add less to make a looser mesh, which is better for separating bigger things. But if you're doing really big things, then you typically go to the Agarose. And so polyacrylamide gel electrophoresis, we often use it in the form of SDS-PAGE, which is where we're separating proteins based on their size. We can also use like urea-PAGE, which separates urea-coated RNA by size. We'll go way into more of this in a minute. This can also be DNA. Or you can also do non-denaturing formats where you're still separating these DNA or RNA by size using one of these PAGE gels. And you can also do non-denaturing protein or non-denaturing DNA. So by denaturing, what I mean is that when we're separating things going through the gel, we can separate them based on their like shape, which would be if we don't unfold them, if we don't denature them. But typically what we want to do is separate them by their like length. This kind of evens the playing field between proteins or between DNA or RNA structures that might have like really strong secondary structures, so they're all folded up, versus ones that are linear, so that you can more easily tell them apart, because typically what you know about this The protein, or what you know about the DNA, is its length. So the number of amino acids or the number of bases. And so you want to kind of erase that shape so that you can separate them just based on their length. So typically when we're doing some sort of like agarose gel for DNA, we don't really have to worry about secondary structure too much. But when we're doing RNA, RNA likes to fold up into these complex like secondary structure states. So the RNA, it has this 2' OH that DNA doesn't have. This allows it to form more kinds of bonds with itself and allows it to form these secondary structures. So basically the primary structure is like a single strand and like just like the sequence, and then the secondary structures when it like folds up, and so it can fold up on itself and form hairpins and things. They're going to cause it to travel weird through the gel. And so we can erase the secondary structure using denaturing agents. And so in a urea-PAGE gel, what we're doing is we're using urea as well as formamide to help denature the RNA secondary structure. It also can denature DNA. And this way is going to allow it to separate more based on just its length. Note that for the DNA and for the RNA, you already have a negatively charged backbone, so you don't have to worry about adding a negative charge to get it to run through the gel. When we're talking about proteins, when we're doing a denaturing gel with proteins, so denaturing, remember, we're unfolding the protein. Well, we're going to need to add a negative charge and we're going to need to unfold it. And so we can do both of these things with SDS, so sodium didecyl sulfate. What it's going to do is it's this detergent, so it's like the soapy molecule. It has this long lipidy tail, so this long like oily, fatty, greasy, blobby stuff, and this negatively charged head. And so what's going to happen is that when you heat up the protein, you help the SDS out. What you're going to do is the protein starts to denature and the SDS kind of sneaks in there. It's going to put its globby hydrophobic tail, so this like water avoided tail, with the hydrophobic parts of the protein. So there's parts of the protein that are normally hidden tightly in the center of the protein away from the water. When you're heating them up, when you're denaturing them, now they're going to be exposed. This would cause the proteins to all clump up and stuff, except that the SDS is going to coat them. And then it also has that negative charge. So it's going to be keeping this protein soluble, hydrophilic, water loved, and negatively charged. So this is going to help it get through the gel. So these are the ways that we can like erase the shape of these molecules if we want to do a denaturing gel. Also non-denaturing gels where we keep things together. So in the case of a protein, this could allow you to keep protein complexes together. If you do some sort of native PAGE gel, you can also run non-denaturing gels for nucleic acids. So instead of having like a urea-PAGE, you would just do a plain like TBE-PAGE. And so TBE is the buffer that we'll talk about that we typically use when we're running the gels. So basically we can think about these main types of our gels. We have the agarose gels, which we're using for separating big, typically DNA, but you could also do if you're doing really big RNAs. And then for proteins, we're typically doing an SDS-PAGE or a native PAGE gel if you want the non-denaturing format. For the nucleic acids, for the small pieces, we're typically doing a polyacrylamide gel. And if we want a denaturing, we can do one of these urea gels with the formamide. There are also ways to do denaturing or denaturing gel agarose gels. These typically use like formaldehyde or some other like newer method, like Collex or something. But I've never done one of those forms, but I've done this urea-PAGE a lot, including yesterday. So I thought I'd tell you more about how it works. So just again, these are used typically for smaller pieces. So remember the PAGE gels, those have that tighter mesh than the agarose, a more consistent mesh. So this is really good if you want to separate small fragments, so typically less than 200 base pairs or so. And it's also really good if you want to can be helpful if you want to purify those out. And so in terms of the actual base pairs, you can vary the percentage of the gel, as I mentioned, so a higher percentage if you want to separate smaller things, and a larger percentage if you want to separate bigger things. And yes. So I know that I mentioned just like that RNA is this more crucial for RNA that you keep it denatured because DNA doesn't really form those like secondary structures as much. But I would mention that when we talk about agarose gels, it is important to keep in mind that DNA, especially when you have a plasmid, it can form like it can kind of coil up and various things like that that can influence how it moves through the gel. And so I have a post on that as well if people are interested. Let's talk about this urea-PAGE. Because with RNA, we really do have to worry about these secondary structures. And even if we're doing DNA or something, if we want to keep the strands separated, then we would want to denature them. And so what do we mean by denaturing? Well, when we talked about proteins, it was unfolding the proteins. And when we talk about denaturing here, it's unfolding strands, any secondary structure, and also keeping the strands apart from one another. How it does this is it needs to disrupt the bonds that are normally keeping those structures together. And the type of bonds that are keeping those structures together are hydrogen bonds. And so they're not strong bonds like the bonds that are actually keeping all the letters together. Instead, these are just partial charge attractions. And so we have hydrogen bonding between the strands. It involves this partial positive charge on the nitrogen of some of these nitrogenous bases, as well as then you have the partial negative charge on the oxygen of bases. And so you can see that each of the four bases, so you have GATC and DNA, GAUC and RNA, and these all have hydrogen bond donors, so the oxygens, and acceptors, the hydrogens on those nitrogens. And so because they each have a combination, they can form bonds with one another. With RNA, you have the additional OH in the extra leg that can also form bonds. And again, these are just partial charge attractions. So they're able to be, you can break them without actually like fragmenting your chains. And so if we replace those hydrogen bonds that it was forming with another strand or with part of itself, another part of the same strand with hydrogen bonds to one of these denaturing molecules like formamide or urea, then we can keep those structured, we can keep the strands like strandy and not have to worry about their shape influencing their run. And so how we do this is first, like we did with the SDS-PAGE, we have to heat things up to allow these molecules to access the sites. And so when you add heat, those atoms that were forming these connections, now they're going to be vibrating, they're going to move apart and unstick. You talk about the TM, which is like the melting temperature, which talks about like how hot you have to get it in order for them to come apart. The stronger the secondary structure, the stronger the base pairing, the higher the TM is going to be. Typically what we do is we heat the samples up for like 90 seconds or so, and at like 80 degrees Celsius. You don't want to get them too hot, especially if you're going to be purifying these out. You don't want to be actually harming the molecules. So you heat them up, and then in your buffer loading, when you're heating them, you have the buffer, the sample loading die. And so typically the sample loading die is going to have, like for the urea-PAGE gels I'm doing, this gel is going to have a few things. It's going to have formamide. It's going to have, which is the denaturing agent. It's going to have bromophenol blue, which is going to be a tracking dye so that we can just see the progress through the gel. We're not going to be actually able to visualize the RNA as it's moving through the gel, but we can see this dye, and this dye is going to allow us to visualize it. You can also see that for this dye, it has different loading buffers. Sometimes you have multiple dyes in there. Sometimes you only want a single dye though. What you want to do is that you want to know what size of RNA or DNA you're looking to see. And you want to make sure that your marker dye is not going to, the dye is not going to run where your size is, where your piece is running. Basically, you don't want the dye to get in the way of visualizing your sample. So you might want to only use certain dyes. So that'll depend on what you're trying to look at. And it also will have EDTA, which we'll talk about more, which is this chelating agent, which basically it hides metals that some of the endonucleases need to chew up DNA or RNA. And we'll get much more into EDTA in a minute. But so going back to this formamide, the formamide is going to help with the initial denaturing in your sample buffer. But what's going to happen is that when you're running the gel, these can kind of like wear off, like because if you only have formamide in your sample loading buffer, now when the gel is running through, you have this like there's no formamide anymore, and so the DNA or RNA would be able to pull back up. You have some help in that the gel is going to get a little warm, but you don't want it to get too warm because then that's going to it can cause your damage to the DNA or the RNA. And so what you do is you put urea in the gel itself. This urea is going to do a similar thing, going to keep denaturing, holding bonds together throughout the PAGE gel. So speaking of throughout the gel, what is the buffer that we're going to use? So typically with these PAGE gels, we're using a TBE buffer. TBE stands for tris borate EDTA. You might also hear of TAE, tris acetate EDTA. We typically use TAE when we're doing like Agarose gel electrophoresis. It's really good for separating large things. This TBE is better for separating small things. So they both have this tris and this EDTA, and then they differ in TAE has acetate and TBE has borate. So what are these various things? So the EDTA we mentioned before. So this is going to be our chelating agent. So a chelator is something that like binds to metals. Chelate like a claw. It's going to bite down on this metal, and it's going to hide the metal. This can hide this metal from endonucleases, which are going to chew up DNA or RNA. So, but this can help you prevent your sample from getting chewed up as it goes through the gel. However, a crucial thing is that RNase A, one of the main RNases that we care about when it comes to like RNA damage, because this RNA is everywhere, like bacteria and other microbes and as well as us and stuff. We make this RNase so that we can basically destroy viral RNA and stuff before it has a chance to get to us. So it's like secreted or whatever. And so it's basically everywhere, and it's really hard to destroy. And so I have a whole post on RNase decontamination methods if you're interested, but basically you want to make sure that you're cleaning all your surfaces really well, and that you're using good sterile technique, filtered tips, filtered buffer, all that sort of stuff to keep the RNAs away, because the RNAs, you can see, it's actually, it gets the RNA to use its OH to attack itself. And so it's not relying on the metal. It's just needs some water to help it out. And so it's not going to be inhibited by this EDTA. But other ones are, so it's important to have that EDTA in there. Okay, and then we have the tris acetate or tris borate. So the tris part's the same. Tris is this pH buffer. The pH is a measure of proton availability. The more protons you have, the lower the pH, the more acidic. The fewer protons you have, the higher the pH, the more basic. Yeah, it's confusing because it's like an inverse log scale. But anyway, so the concentration of protons is going to determine the pH, and what determines the concentration of protons is going to be the amounts of like acids and bases in there. A pH buffering agent is able to act as both an acid and a base, and so tris is one of these pH buffers that it can donate or accept protons depending on the pH of the solution. And so with this weak acid, the boric acid or the acetic acid, that's going to act as a source of protons to kind of help get that initial balance going in that proton, the amount of protons, so that the tris can then like keep the pH steady. So this is going to be your acid form conjugate, like complementing the tris base form. And so the borate is just going to be the form of boric acid that has given up a proton, and then the acetic acetate, the acetate would be the acetic acid that has given up a proton. So you might see both of those terms used. TBE is, this is a better buffering capacity, so it's better for doing like the long runs that are important for separating like small things like PAGE gels and stuff. Typically, these gels are going to be run for a couple hours, even for those small gels, even longer for the bigger gels. If they're going to be like high voltage and stuff, what's going to happen is because you have that tight mesh, it's going to take more energy and more time for the molecules to travel through than when you were running one of those like Agarose slabs. The TAE This doesn't have quite as good of buffering capacity, but your runs are shorter. And crucially, it doesn't have the borate or the boric acid, which is basically, it can inhibit some things like ligases. It can make it harder to extract the DNA out of the gel, stuff like that. So typically we use the DEA, and we also, the resolution is also better for the bigger things, and this is better for the smaller things and stuff like that. So yeah, so DEA, we typically use TBE for these sort of gels in the gel running system. So that's what's in the gel running system. So how do we actually set up these gels? So you can cast your own, you can make your own, or you can buy pre-made ones. So yesterday I was running one of these pre-made ones. So you can see this is like a 15% TBE urea gel. And you can also, in my old lab, we made our own. So we can use we use this like urea-seq gel system. With these different components that you can mix and match to get the percentage of gel that you want. Because remember, the percentage of gel is going to influence the size of the mesh and therefore whether it's better for separating smaller or bigger things. When you make your own, you can also make all sorts of different sizes of these gels. But yesterday I was just running one of these little guys. So as I mentioned, you want to heat up your sample when it's in the loading buffer, but you're not going to overheat it or anything like this. You're not even going to do this until you started pre-running your gel. So you want to run it in the absence of any sort of sample, so that you can get the gel to have an even temperature throughout, or else what's going to happen is that parts of the gel are going to run faster, or parts are going to run slower. And as I mentioned yesterday, you don't have like a stacking gel or stuff like that. So everything is going to, you don't have things concentrated down to the line before they go through. So basically when you're pipetting in, if you have stuff at the top of the column, it's not going to concentrate into a nice sharp band. You're going to get more of a smear. And so another important thing is that you actually clean out your wells so you can go through and you can wash the different wells. So you can go through and you can actually watch the different wells to make sure, like, because the urea can kind of like leach out and you get this kind of more viscous layer in the bottom of the well. You don't want that to be there. That can interfere with the run of the samples. So you can also get little gel chunks in there and stuff. And so you can use a syringe or a pipette tip to like pipette up and down and wash the wells. And you can also do that like right before you load a sample. Just make sure you're not like squirting stuff into the sample next to you. Then it's time to load your samples. I have more yesterday about good techniques for helping you load the gels. So you want to pull up, make sure you don't have any bubbles, and then pipette directly in and pull up with your thumb. And so more on that in yesterday's post. You want to work quickly so that when you're loading, so the samples don't diffuse. So you can see that as I loaded these, the samples are already kind of starting diffusing. When you're loading the samples also, much more in yesterday's post, but you want to make sure that you're loading things in all of the wells. So if you don't have enough samples to fill all of the wells, you want to put 1x sample buffer. So if you have like a 2x sample loading buffer, you can just make some 1x by mixing it with just like tris or TBE or whatever. So you have a sample. This will keep the current constant throughout all of them. You want to put your samples in the center, because what's going to happen is you typically get this like smiling effect where things in the center are going to run faster than things on the edge. And you get this kind of like things on the edge kind of get pinched together. And so you're going to get better resolution and better able to tell things apart in the center region. I also usually put a ladder on each side in case you get funny things running funny, as well as a ladder in the middle if you have enough room, so that because of the whole like smiling effect and stuff. Also, what I do is if I have empty lanes, remember you want to fill those empty lanes, but typically I fill them last, so that if I have any sort of loading problems when loading, I will have extra lanes that I haven't wasted with the sample buffer yet. So I will count where I need to calculate out where I need to start with my samples, put my samples in, and then add the blank lanes. You can see that once you start the gel, you should see little bubbles forming, and then you'll see that the bands you can start to see the bands are going down into the gel. Typically, you run these for a while. For the big gels, they can get really hot, and you need to make sure that they're not get too hot. So if they get too hot, they can crack. So you can see this one even cracked, even though it had this, we had the metal thing on it to try to keep its temperature steady, and we had a little like temperature monitor. But yeah, so these big ones can be a pain. These little ones are better for that. Okay, so what you're seeing here is just the sample loading dye. So in this case, it's bromophenol blue. And so this dye is visible to us because it's stealing part of the visible light spectrum, and much more on this in other posts. But we can't see the RNA or the DNA because although those absorb UV light, they don't, we don't see when the UV light is stolen. So basically when visible light we see, because part of the like RGB, like the whole like rainbow, the white light is made up of all these different colors. If you steal some of those colors, so if something absorbs one of those colors, then you see a color. But in the color that's the opposite in the color wheel. But when something steals invisible light, like UV light, then we don't see it. But we can use techniques, we can use other dyes and machines to help us see things that absorb UV light. And so although DNA and RNA can absorb some UV light themselves, we typically add some sort of fluorescent dye. Something, so for like DNA, we're typically using some sort of like DAPI or ethidium bromide or some sort of derivative. So when I do my RNA gels, I typically stain them with this cyber gold nucleic acid stain. Basically how these work is they involve fluorescence, which means that you shine a wavelength of one light at them, and they're going to absorb that light and then give off light at a different wavelength. And so you can measure this with the machine. And I have much more fluorescence in other posts. I'm not going to get into it, but you can use one of these gel dock machines. What can be helpful is what you can do is if you're going to be like cutting your gel off, you can stick some saran wrap in like in whatever gel dyeing box you're going to use. Remember, if you're doing this for RNA or something, you want to make sure it's a very clean gel box, so you can clean it with like some detergent, and then some ethanol, and then some really clean water. But what you want to do is you can use some sort of dish if it's a bigger one. What I found you could do is you put saran wrap in it and then put the stain on top of it, and then put your gel on that, and then you can actually transfer this, just take it out on the saran wrap, so you don't have to worry about tearing it. And so yeah, that's good if you want to then extract things out and that sort of thing. Just to know, the cyber gold it's like a 10,000x. So if you want to say make 50 mils worth, that would be 5 microliters, and you can make little aliquots of it and store those 5 microliter aliquots or something. So that's what they do in this lab, and it's really smart because those bottles it like freezes, and then it's really annoying to try to thaw it because it has to be shielded from light and various things like that. Okay, you can also use radioactive methods. So in my old lab, I did a lot where I had radio labeled RNAs, and you can then use these phospher imager screens to actually visualize them based on the radioactivity. Okay, so I think that is the basics. Oh, you can also do blots. So after you run the gel, so this is showing an agarose, you can also do like a northern blot out of the PAGE gel onto a membrane, and then probe with various sequences to look for the presence of those sequences. Okay, so now I think I'm really done. And hope that helps, and happy running your gel.